Hello, this is Hakwan Lau. I'm based at UCLA and also I'm partially affiliated at Hong Kong University. So today I want to talk about consciousness and fear. Thanks very much for inviting me back here. Of course, many of you, I know some of you are old friends. I used to work at Columbia here left about 10 years ago and moved to the West Coast. So good to see so many of you. Looking forward to chatting about what I'm going to talk about next and everything else. So the question I want to ask today is a little bit of an indulgent question, as some of you know that I tend to be a bit like that. And I want to ask a kind of broad question about whether we are alone in the entire universe or animal kingdom, at least, in having the capacity to feel anything, such as fear, joy, sadness, happiness, that sort of stuff. Are we alone in having the capacity to feel that? And some of you might think this is already a bit of a non-starter because on the internet you see videos like this, right? So this is a clip from YouTube. So looks very obvious that the dog was kind of mortified, and probably other people who have animals would say that the animals clearly feel emotions. I think that's right. I think they do look like that. But the problem is we anthropomorphize about these things a lot. So this is a plant called Mimosa pudica, sometimes called a shy plant. And it's being touched. Obviously, it also looks like it shies away, or if you use a bit more imagination, you can think it is kind of being afraid. It is scared by the touching, and that's why it kind of shrinks. And that is, I think very few of us think that the plant really feels emotion. So they kind of brought up the kind of problem. So just because some sort of creatures or animals or even plant looks like that they're reacting like they're like they have emotions, it's not clear whether they truly have the emotional experience. And in fact, Darwin actually wrote about that, and he said like clearly the expressions of emotions are evolutionarily continuous and inherited from our ancestors. But how about the experience itself? And when asked about that, Darwin doesn't really have a good answer. He actually resorted to saying, well, it's more cheerful to think that they are conscious rather than not. And Joe Ledoux, of course, wrote about this too. And it is, I would say, is an unresolved problem. And another way to think about why it's unresolved is to think about the whole spectrum. So obviously, we think we are conscious, and probably many other primates are. What about a cat? Then I think many of us are going to say yes. But if you go down the line and think about a bird, a reptile, a fish, and then shellfish, and then what about bugs? Um, and I think some of us are going to stop somewhere and think maybe a bug is not conscious. But what about a robot? So some people say that you have to be alive to be conscious, and that rules out the robot. But the robot can be a lot more sophisticated than a bug, obviously, these days, at least in the near future, it will be. And then an oyster is alive, and trees are also alive. And then bacteria culture are also alive. And I think on the very end of the spectrum, we have stones, and I think very few people would think stones are conscious. I do have some colleagues who think stones may be conscious. In fact, they think photons may be conscious, but I think you don't necessarily want to talk to them. So where do we draw the line? I think I personally may draw the line somewhere like here, and other people might move it around. But I think it's clear that once you think about the whole spectrum, the absolute position of that cutoff is unclear. And it's very hard to say that we have a good scientific answer for now. So I think there's a way to do it. Is not bulletproof. It's basically by induction. So we try to work out what are the so-called neural activity or neural signature for the capacity of having conscious experience. So we can do it in vision, where experiments are easy to do. So we can present an apple to a person, and the person said, "I saw the apple," press the correct button to indicate the stimulus was an apple. And then you can compare it with another condition. You do some tricks such as like visual masking, whatever you like, TMS or something like, or give the person some drug so that you would block this like channel so the person cannot really see consciously. So presented with an apple, the person might just say, I don't see anything, and fail to press the correct button. And you compare these two conditions, you have what is so-called the neural correlates of consciousness, the NCC. And then you can then go and proceed and check, let's say the NCC is some sort of pattern of neuronal firing in the prefrontal cortex. Then we can go and check whether other animals have such a structure and have these physiological signatures. Then if they do, then you might say, oh, then very likely they're conscious because they share this very same NCC. So it's not a bulletproof mechanism because it's induction, and some other creatures might have brains very different from ours, but it's better than guesswork, I would say. But there's a problem with this approach, and I think that pretty much summarize a problem of the entire field of studies of conscious perception. The issue is when we compare these two conditions I just showed you, in fact, what differs is not just that in one condition you're conscious of the apple, and in fact, your basic ability to process, perceptual signals for the apple is pretty weak in this condition, in the control condition. So there's a confound there in terms of consciousness differ, but just perception also differ. Now then, some people might say, well, consciousness just is perception. There's nothing more. In fact, there's just a science paper that just came out, I think last week, that said that birds are conscious because they can detect stimuli, essentially. That's, I think, a little far. I think the photodiode can also detect stimuli, and we wouldn't say it's conscious. So I think we have to distinguish what is really the subjective experience from just the mere having of the perceptual signal. So in other words, there's a confound here. The person not just cannot see the stimulus, also cannot press the correct button. So with this kind of task, I mean, we've done it for basically over 2 decades now. And the answer is kind of the same and I think not necessarily very interesting. Essentially, the NCC is everywhere. When you compare this condition with a very huge, very intact perceptual signal versus a small or negligible, non-existent perceptual signal, you basically activate pretty much the whole brain. I mean, not the whole brain, but all the brain areas that are related to vision, basically from B1 all the way up to dorsolateral prefrontal cortex, etc. You find it everywhere. And the signal, of course, is also strongest in the sensory areas because you have a huge signal difference in vision. In this case, basically, there is intact vision. In this case, you have impaired vision. So it may not be so clear that this really has anything to do with consciousness per se. But this is pretty much a problem in, I would say, over 99% of the papers you read that has the word consciousness in the title or the abstract. Most of those studies, it has basically, I'm not sure they really are about consciousness per se. So I think there's a solution to this if you follow the diagram. On my screen is a little cut off here, but this is just the same diagram earlier here, showing that the condition, the unconscious condition, when you see the apple, the person might fail to press the correct button. So usually you compare these two, but I suggest that you replace this condition with something like this. So here you present the apple to the person would tell you that I do not consciously see the apple. But if you ask the person to press the button anyway to indicate what it is, it would have a higher than chance likelihood of getting it right. So that is, it seems like there would be some sort of non-conscious signal that is quite strong, and yet the person does not consciously see the thing. So this is basically the classical neurological condition of blind sight. So if you compare these two, then there's a chance that you can actually match the signal strength from here entering to here. Basically, they can be equally strong in terms of your signal detection theoretic measure like D prime. Your performance can be both at the same level. Let's say you give it a slightly weaker signal, but still very conscious. So your performance might be at threshold. This one performance will be at threshold. But in this case, you would see the stimulus. In this case, you don't. If you find that, then you can get rid of this signal or performance confound. Then you will find the true NCC. We have done something like that a long time ago when I was a young postdoc, actually. So we did exactly what I was told. So what I just told you, We found a patient, GY, who's a blind sight patient with half of his visual field damage. So the lesion was on the left V1, uh, and that means that on his right visual field is subjectively blind. Then you can present stimulus to the blind field and to the good field. And as I said, you can titrate both. So you can make the stimulus slightly weaker on the normal side. So the performance is exactly the same. So this is a simple 2A FC task. They have to tell the spatial arrangement of two grading patterns. And so they're equally able to do both tasks. And yet you can see that in the blind field, basically, they don't see the thing. In a normal field, they don't say that they see the stimulus 100% of the time either. In fact, so the subject saw the stimulus less than 50% of the time because it's near threshold. So sometimes he would say, I didn't see. In the blind field, he saw a few trials. And then the reason is we encourage him to report any kind of awareness. And in fact, sometimes it's like light scatter from the normal side. He saw some sort of flicker. But if you ask him, he really never saw them clearly. And if you ask him to also bet on which are the trials he's likely to be correct, on the normal field, he's pretty good at doing that. In the blind field, he really had no clue. His bets were kind of all over the place. So that's like one study that we've done. That seems to work. And you compare these two conditions, then what you found is that you find this frontal and parietal activity preserved. So these areas are pretty commonly activated in normal studies of conscious perception. But in this one, it's kind of interesting. It really even survived this kind of control, even when performance was matched. Comparing these two conditions, you still find it. And you can do similar studies in normal subjects too, maybe undergrads. Basically, they are subjects from the general population, and you can use psychophysics to achieve a mild version of this. So these are not brain damaged patients. So when performance is matched, you get only a small difference in the subjective ratings of how often they see the thing. This is using a technique called meta contrast masking. I'm not going to here because going to here is a really old study, but it basically found something very similar. You only found activity in the prefrontal cortex in this case, and pretty much nothing in the visual cortex. Now then, I don't think you really wouldn't find anything in the visual cortex if you put electrodes in and use very sensitive invasive measures, given that this is a visual manipulation leading to a subjective visual difference. I think chances are you also find something in the visual cortex. The stimulus are not completely matched. But I think it's quite interesting that you find prefrontal activity at least more salient in this case, because usually your sensitivity for the visual cortex is much higher. You can do retinotopic mapping and basically register the signal really clearly. In prefrontal cortex, it's kind of surprising that we found something even under these controls. So that kind of led people like myself to think maybe the true NCC is really in the prefrontal cortex, in particular dorsolateral prefrontal cortex and frontal pole, that is areas around here and here. Okay. So many people, we've been saying that this prefrontal NCC idea, we've been saying it for some time. Some people are not happy about it. Some people think it's completely wrong. And the reason is mostly because of lesion. So the reason is if you lesion the prefrontal cortex, people don't seem to go blind. But it's actually a very tricky issue because lesions are usually, I mean, unless in monkeys, you can really prepare your lesions. Normal human subject lesions are due to stroke or trauma, then they are typically unilateral. And we know that the prefrontal cortex doesn't quite work like V1. It's not like you lesion out one part, then the other side, the contralateral side is gone. It works much more bilaterally. So if you lesion one side, there is evidence that the other side would just take over. So it's hard to assess how it goes. And when you have truly bilateral patients, then they tend to be very hard to be tested. In fact, they tend to be very impaired. I can show you a video later, but I proceed to tell you how do we address this problem. Because in monkeys, it's hard to ask the monkey whether you're truly conscious, because we don't think that it's going to impair their visual performance. We think that it's going to impair the subjective visual experience. The performance might well be intact. So we use theta burst TMS. We send TMS to kind of transiently depress the activity of an area with a protocol called theta burst. Basically, it's a trained of gamma modulated by theta rhythm. Then by TMSing about 1 minute, you can basically depress, reduce the activity, the responsiveness of a piece of cortex by up to an hour. So then you can TMS one side and wait a little bit and TMS the other side. So you don't have to concurrently TMS both sides. So it's relatively safe in terms of seizure induction, but you can lead to a bilateral mild impairment. And in fact, it does do something to the subjective awareness. So this is a sham condition. You see the pre-post, so is a pre-post real sham design. So before TMS and after TMS, so just showing that it's not due to fatigue that there's a change, but if you actually use real TMS to the prefrontal cortex, then a certain measure drop by about 20 to 30%. So what is this certain measure? And this is actually a measure developed by myself and my former student at Columbia. And essentially is a measure of how your accuracy, how your trial by trial accuracy in doing the task. So again, it's a simple spatial 2AFC task. How does that accuracy get correlated with your subjective rating? That is, usually people who say that I saw the stimulus, they tend to be correct. And when they say they don't see the stimulus, they are less likely to be correct and more likely a chance. So there should be a correlation between these two things, right? So you can just run a correlation, and some people do that. But we actually translated it back into a signal detection theoretic scale. We call it meta D prime, so that it can be compared with D prime. So then we can say, so basically D prime is your signal to noise ratio unit your information signal to noise ratio unit and available for you to do the perceptual task. And your meta D prime is the same measure, but it's the information available for you to rate this subjective rating meaningfully. So basically, you can say how much for the information you have, how much information is available for your metacognitive disposal or metacognitive access. So that dropped by over 20%, and for a TMS effect, is actually not tiny. I mean, it's pretty decent. So we were happy about that. And if you extend that a little bit, you realize that you don't have to ask something so nebulous or weird as subjective experience. I think it's not too weird, but some people want to ask something more objectively. You can ask them about confidence. So after they do the task, they rate they gave the 2AFC judgment. You can ask them how sure you are that you're correct. You can ask them to bet money, etc. And if you do that, then you can again calculate this M ratio, this meta D prime divided by D prime. And Steve Fleming in London has done study like that. In fact, he did it when he was in New York, when he was at NYU. And he looked at patients. So now he actually went to the unilaterally prefrontal lesion patients and then tried to do these tasks. The idea is, well, maybe they don't complain about losing consciousness because it's not 100% knockout, but there might still be some effect. And in fact, he found that this M ratio measure changed by about 50%. So it's actually a pretty solid effect. The problem is, prefrontal patient lesions sometimes would lack insight, so they may not be able to tell you that the vision now is somewhat problematic. But also the visual performance didn't change. The D prime didn't change. They perform visual tasks just as well, even at near threshold. It's just that their introspective access has become poorer. And also, Steve ran this study and tested them on a memory task and showed that there is no impairment. So it's not just that in general they become less introspective or become less able to do these fancy, self-monitoring tasks. It is specific to perception. They are less able to monitor their perceptual abilities. And others have done it in monkeys. So there's a monkey paper in Science, and they injected muscimol into prefrontal cortex. And again, it impaired this measure very, very strongly. And there was also a rodent paper, I think, in rats, and they again injected muscimol into orbitofrontal cortex, and again find a huge knockout effect on this kind of metacognitive measures, but not the first order D prime. Not so that the rodents are just as able to do the odor discrimination task. And if you still you think these are metacognition, as I said, is specific to perception. But if you really don't buy that, there's also some evidence that prefrontal lesion changes your detection bias. It makes people more conservative in reporting that they see something. All of these are near threshold effects. I understand it's not like complete knockout, but at the same time, these are pretty strong converging evidence, I would say. And then some other people are still not happy. They think that all these prefrontal activity doesn't really reflect their subjective experience, but it only reflects their report. I'm not going to go into that because some of these people are also people who believe that, stones and electrons might be conscious. So maybe we can put that aside. And I have already reviewed that in another review paper addressing some of these. And also later on in the talk, I'll give you one piece of evidence why it cannot just be report. It is more complicated than that. Okay. So you might think like, why PFC? So I hope I convinced you at least it's worth thinking that it might be PFC related. The reason is this. I think you can theoretically think about why PFC is important. So let's say you see a duck. So you have a, first order sensory duck neurons firing. I mean, they're not called duck neurons, but you know what I mean. They're neurons in the ventral stream that code for specific objects and animals. And these neurons can fire for a number of reasons. So if you're just imagining a duck, then you probably would activate these neurons. At least some neuronal activity would be there in some ways, probably in the form of local field potentials. And of course, the neurons also spontaneously fire. I mean, no neurons are perfect on-off, and even when you're doing nothing to the animal, or even when the animal is partially anesthetized, there's still a fair spontaneous activity, in fact, that accounts for activity. And of course, there's another scenario, which is that you're actually seeing a duck. So this duck neuronal activity is reflecting, is a legit reflection of the state of the world right now. So you have these three different scenarios. And I'm not saying that the neural activity in the visual cortex are exactly the same in these three cases, but they're quite similar. You can use the same decoder and decode them with fMRI, ECOG data, etc. So it's plausible that, it's at least possible that there is a downstream mechanism to distinguish between these cases. And if you think about that mechanism, then that mechanism is kind of like a light switch for your consciousness. Because if there is such a mechanism, then given the same duck representation, if I change from this mode to saying that there is an external duck right there, and to change it to say, no, actually, this is just internal noise, then you should not be seeing the duck because we don't experience our internal spontaneous activity. At least not directly. The phenomenology should be very different. And likewise, if you're holding the duck online, some people have very strong visual imagery and some people don't. So by changing about these cases and this decision mechanism, your phenomenology should change. And that might be why PFC plays such an important role. And I've expanded on this idea elsewhere in the preprint, so I'll not say too much more. There's a philosophical literature behind, but let's talk about the science. So Diego Mendoza Holiday have in fact published a paper showing that in the PFC, there are neurons that exactly distinguish between internally held representations, they're doing dot motion in monkeys, versus actually perceived stimulus that is concurrently on the screen. So there are neurons in the prefrontal cortex that can read out the direction of the stimulus, but there are different populations for the external perception case or the internal memory case. So you have some evidence that there is something like this in the prefrontal cortex. And then I already show you the other case, which is the perception case and the noise case, which basically is the metacognition task that we talk about. If you think that what you see is just noise, then you rate low confidence and you might be in a blindsight-like situation, etc. And then Sam Gershman also have written out the math, and there is a kind of engineering argument why you would like to have something like this in your prefrontal cortex. Essentially, that's taking on a concept from deep learning, which Ian LaCoon, another of our New York colleague, has called the coolest trick in deep learning in the past decade or so. The idea that if you want to train a generator, you want to train predictive coding network, then the best way for you to do it is to train something like that. It's called a discriminator to challenge the generative models. When you do top-down generation, you want to create a discriminator to distinguish between these two cases to see whether your internal generations are close enough to the real thing. And then you set up this network to compete with your generative network. Then the two networks will be trained very well. Again, the math is like all there, but the idea has been very influential in AI in general in the past decade. So there's an engineering argument why we might want to have something like that in the prefrontal cortex. So let's say it's true. There is a bit of philosophy that is interesting too, because it also explains why when you consciously see something, you kind of see that it's almost like a logical proof that there is something. When I say, why do you think there's a duck? You say, well, I see it. Why does it give you this certainty? And there is some sort of philosophical reason why you think this might be good to have two separate representations for that. But I recommend you to go to my preprint. Let's not get into too much, or maybe we can go further in Q&A, because I think probably not many of you would care about this sort of stuff, but it answers the old philosophical conundrum. I think it's kind of interesting. Okay, so now let's see how this model applies back to emotion. So I started out by asking about fear and joy, happiness, all these more important things than seeing a Gabor patch. So does this theory, or at least the PFC idea of the NCC being in the prefrontal cortex, does this idea generalize to emotions? And I think it does, and there are quite a bit of evidence, but I'm going to show one study done by ourselves. Vincent Castro de Moschel is a postdoc in my lab, and last year published a paper in which he, in the studies, he presented a lot of stimuli to subjects, and the stimuli are from different animals and some man-made objects, and people just have to detect an immediate category change. So very simple task, very fast. So it's like snake, 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 and then change to a rabbit, then you have to press a button. And basically we just presented a lot of animal and objects, images to people, and such that we can build decoders and decode them, etc. But one thing that's quite interesting is your people's subjective fear rating. So we can also ask them how these animals scare them, and a rabbit is like mostly 0, a snake might be some. And then you can also measure their physiological arousal. And it turns out, of course, there's a correlation. But the correlation is not perfect, and in fact, it's quite systematically not perfect. So it's not like just noise that it falls out. Some people just do not subjectively say they are so much afraid of spiders, but when they see one, their physiology is like raising very fast, and sometimes the opposite happens. And so capitalizing on this fact, there's a bit of dissociation. So that means that we can build brain decoders that would differentially decode the two categories, meaning the following. So in the mostly prefrontal cortex, you take these voxels and you try to do multivariate voxel decoding, which I think I don't have to explain because Nicholas is a colleague here at Columbia, and he really pioneered a lot of this stuff. So I assume most of us know this by now. So you can take multivoxel from certain regions and try to predict the subject's subjective rating of fear. And you can do this pretty well in prefrontal cortex. And yet, these same areas do not actually decode the physiological correlate of fear or threat nearly as well. In fact, they don't do so well at all. And then the opposite is also true for the other areas like amygdala, insula, ventromedial, prefrontal cortex. Typically, the areas associated with like limbic, emotional processing. They are kind of the opposite. You decode the skin conductance and all the physiological drive pretty well, but it doesn't actually decode the subjective fear rating nearly as well. So there's a dissociation. And that seems to go well with the idea that your amygdala, insula, EMPFC might be like the visual cortex in the visual NCC case. So they correlate with the the fear or the threat signal, the raw signal. But for that signal to be really conscious, you need this higher order prefrontal mechanism to really translate that into the subjective rating of fear. Okay. And earlier I said I want to address this report problem. And so here we are. The issue is you have what during the task, as I told you, people were just detecting category change. They're not rating fear. In fact, fear was rated outside of the scanner after the experiment was done. But the fMRI data collected during this procedure nonetheless predicted the subjective rating of fear subsequently. And that is my point about like this may not be truly, this cannot really be reporting, just reflecting the report. So they are not reporting the fear during this, while they watch this, but their subsequently reported experience of fear for the categories are nonetheless predictable by their, from their prefrontal patterns. Okay, so I finally moved to the very last part of my talk. Hopefully I would not overrun. So the above might challenge an idea that many people find odd, because in textbooks, people would say that amygdala is the fear center. And of course, some of you know that there's a pretty big debate going on with Joe claiming that this is not quite right. You should not call it a fear center, you should call it a threat. I leave you to debate with Joe. I am more or less on his side in the sense that I think amygdala certainly is very important for fear. And I think Joe would agree with me too. But it's just that the signal itself, in the absence of any prefrontal mechanism, may not be sufficient for the experience of self, of fear. It might more be driving your physiology, which obviously is also an important part of fear. You can call it threat processing if you like. And there's some evidence in support of that. There's some lesion evidence. If you lesion the amygdala, people can fear. And then there from direct stimulation. When you stimulate the amygdala, actually very few people feel fear. I mean, sometimes you can at a very high intensity, but I suspect the reason exactly is you have to stimulate the amygdala strong enough so that the activity would leak, would further go to prefrontal cortex. That's when you actually feel the fear. Otherwise, it just drives your physiology. But to test this idea, since it's so odd and so relatively new, I think it would be good to have a way to show that you can actually change amygdala activity using a more systematic way, not just stimulation in a few subjects, but to maybe do kind of threat conditioning, that is fear conditioning, but this unconscious, and to have it only changing amygdala activity, but not your subjective experience. So that's a mouthful. I will explain to you what the study is. It kind of tests the idea that amygdala may just reflect unconscious threat processing. So it's actually a study using a technique that is called Decoder neurofeedback, and it's really invented or pioneered by my colleagues in Japan. And the idea is you basically get people to do something a bit like neurofeedback. So you do online real-time fMRI imaging. And after you tell people, okay, focus on your mind for a little bit, and then you decode the brain pattern, and then you just give them a feedback. If their brain pattern look like what we want them to look like, then you give them a big feedback, and then they earn about $0.1 or something. And then you repeat for a few hundred times. So you basically have to be done over a few days. A few sessions spread out over a few days. And the advantage of this is this technique is something called DECNEF, standing for Decoded Neurofeedback. And the advantage of this is people have no idea what goes on. So you don't even, so this is why it's unlike neurofeedback. In neurofeedback, usually you explicitly instruct people and tell them what to do. But here you don't. You just tell them, just do this mental slot machine, I don't care. And if you do the right thing, you earn money. And by the time they come out and then they earn a fair bit of money, you ask them, so what were you doing? What's your strategy? The strategies are tend to be completely unrelated to what the actual pattern target is. So they really have no idea. And I think that in the beginning, I think we were quite puzzled, but the more we think about it, it makes sense, because really, I think we're just rewarding and trying to reinforce the spontaneous occurrence of fMRI activity. And when the pattern is right, we just reinforce it so that it happens more and then to get paired with whatever stimulus presented here. but it really, you don't feel your unconscious spontaneous activity. When your duck neurons fire every few seconds, you don't see a duck. So you have a way to unconsciously pair or activate a voxel pattern and pair with money. So here's the logic of the study. We, using the same data set, actually Vincent collected, we can decode quite easily two things that people are afraid, and we can tailor-made choose these two things for a subject. So it can be they're afraid of snake or spider. And then, okay, you choose one of them to become your target, and the other one becomes the control. And you randomize double-blinded. I would explain why this is cool in a bit, but it's completely computerized. And then you choose, you make that the target pattern. And then through this neurofeedback procedure, every time this thing happens, let's say this is a target, the spider happens, you give them a bit of money, I mean, via the signal. So over time, they should actually be counter-conditioned to like this thing, because now this thing, they start off being very negative and very scary. But over time, every time this voxel pattern happens, they get a bit of money. So maybe the idea is then over time, they would come to like this rather than hate this. And it turns out it's true. So, and this is the amygdala bold activity for when they actually subsequently represent those pictures to them. This is for the active condition. That is for, let's say, the spider just now, which went through this counter conditioning procedure in the brain. And you can see that the amygdala is completely gone. I mean, the response, they're no longer much afraid. And the control remains just as strong before and after. So it's specific to the voxel pattern, specific to the category of animals that they're afraid of. And this is the same thing just for skin conductance. It's a bit noisier. You might think this is kind of too good to be true, but in fact, we have seen it once already. We've seen it once already in another study published earlier. And in fact, when we saw that first study, we didn't quite believe it. And it was actually just a more simple study. It tried to condition out condition fear, which we similarly pair with electric shock. And then using the same procedure, we also eliminated fear for a specific target. And here is more like naturally occurring fear, and that's kind of cool. But here's the most relevant part. In fact, the subjective ratings of fear, again, this is for the target and for the control, you can see that it doesn't change at all. So this non-conscious counter-conditioning or reinforcement procedure flattened the amygdala response and the skin conductance. But you ask people afterwards, did you, now you see these stimuli, how afraid you are about these things? They would say that it's just the same. So we thought this is kind of cool because we have now an unconscious way to do clinical psychology directly in the brain. And we, of course, we know that my colleagues, such as my colleague, Michelle Krask, there are many experts on this kind of clinical treatment. Essentially, people are afraid of something, if they're phobia or PTSD, the way is for them to re-expose to the experience in a safe setting. So overall, they condition themselves to stop to be afraid of the stimulus. But of course, people are, the major problem of the treatment is people just leave. They tend not to complete the treatment. Because it's quite unpleasant. I mean, if I'm afraid of spiders, you tell me to hang out with spiders. I know that it might help, but I do not like doing that. And the other issue is relapse. So there are some challenges with the current clinical procedure, but if you can do it in the brain and non-consciously, obviously people then do not even have to think about the spider. I mean, all of this just happens in the brain. It's like putting your head into a washing machine, then presumably they would not find it so hard to go through and will help with attrition. And also because it's directly on the neural representations, there are some arguments to be made that it might be context non-specific. So it will be, you just go to the source, so it might help with relapse. But it kind of brings us to a problem, how do we decode those things in the first place? So if you have to present a lot of spiders to people to decode, it wouldn't work. But there's one thing really cool about the paper, which is we made use of a technique called hyperalignment. So essentially the idea is I present to you dogs and cats and butterflies and whatnot. And then I find a surrogate, a subject that also saw these dogs and cats and butterfly. And then I put you two in the same space. And once you're in the same space, then the surrogate can go and see those spiders and snakes and all those scary things that you don't want to see for you. And then I can, from that, infer what your spider and snake patterns would be. So someone else can, tolerate the stimulus if they are not so phobic. to those stimuli, and then I can infer your voxel pattern. And you might think this is too crazy and wouldn't work too well. Turns out it works very well, because once you can do one surrogate, you can do 100. And when, in fact, we have about 100 people who have seen this stimuli. So we are now in sort of pretty big data regime to do this decoding. And so even though you don't have to see something, I can predict your voxel pattern pretty well. And that was already implemented in Vincent's paper back in 2018. So it's still ongoing. We're now right now running a clinical trial. Clinical trial is pretty harsh. Sometimes it works, sometimes it doesn't. But I think even if we fail, I think we're doing something good here, because usually in clinical psychology, it's just impossible because of the nature of the treatment. It's just kind of hard for it to be double-blind controlled. And here we're doing double-blinded clinical psychology. So hopefully we will find out something. So it's time for me to wrap up. I have started off with a kind of crazy question, but I think the answer may not be so crazy. If you follow the logic, we're basically trying to find the NCC within the human brain. And with just the NCC, then it's hard to generalize. So it is also good that we try to understand a bit of theory, why it does what it does and how it contributes to your subjective experience. And I've showed that this, although most of this come from vision, it generalized to emotions too, so it may generalize to more modalities. So as to the question, are we alone? I leave you with this conundrum that I think one of these guys is probably conscious, the other one is extremely delicious, and so you figure out which is which. Thank you. Thank you.